Today, we are going to study about the various models of our plasma membrane. First one is our sandwich model. Alright, so you can see the diagram that there are globular proteins, alright, and there is a phospholipid bilayer. Okay, this is our globular proteins, and here we can see the phospholipid bilayer. Now, let us discuss about the phospholipid bilayer nature. Okay, basically a phospholipid, typical phospholipid molecule is amphipathic in nature. What does it mean? It means it consists of two types of nature. Okay, so here it consists of a head and a tail. Tail is made up of our fatty acid chain, and our head is made up of our glycerol. Alright, our head, which is made up of our glycerol, is hydrophilic. Hydro means water, philic means loving. Okay, it means it attracts our water molecule, but our tail is hydrophobic. It repels our water molecules. It does not attract to it. Alright, so this is the basic two nature of our phospholipid layer. Hence, we called it amphipathic in nature, and this type of nature is given by our Daniel and Dabson. So, first of all, we are going to discuss about some features of our sandwich model. Okay, it is basically given by our Daniel and Dabson. Remember, our sandwich model is the basis of all models given after that model. Alright, and most important, it is basis of our fluid mosaic model, which is we can refer in the modern time. Right now, the first point is sandwich model. According to Daniel Dabson, plasma membrane is made up of our proteins and phospholipids. Okay, as I told you before, it is firstly told by our Daniel and Dabson only. Right. Now, the second is our proteins. He said that proteins are made up of alpha globular molecules. You can see the diagram here. These are in circle shape, right? This is our alpha globular protein, right? Now come to the second part. As I have taught you in the previous section that our phospholipids are amphipathic in nature, right? It means it has one tail and one head. Alright, so it has a tail, it has a head. This is made up of glycerol, and this is made up of fatty acid chains. Right. Now come to the next part. Yeah. Okay. So you can see sometimes phosphorus is also attached to it. We will discuss it in later classes. Now come to the next point. According to the sandwich model, phospholipid is arranged in the form of bilayer. Alright, it is arranged in bilayer form. You can see the diagram. Okay, face each other. Means their tail part are facing each other like this. You can see here in the diagram, their tails are facing each other. So you can see in the bilayer form how they can arrange themselves. They can arrange in such a manner that their tails facing each other and their heads are away from each other. Are you understanding? Alright, you are very good. Now come to the next point. According to him, let's draw the diagram, right? So there is a two. You can see how the bilayer hold each other with the help of a force of attraction, which is our van der Waals force. Okay, and you can see here, they said that proteins are on either side of the bilayer. Okay, you can see in the diagram. Now let's the proper diagram of our sandwich model, which is given by our Daniel and Dabson. Here you can see the bilayer nature, which is holded by van der Waals forces, and there are proteins on either side of the bilayer. Right. Basically, this is the typical structure which is given by our Daniel and Dabson. There are obviously some limitations of this model. Hence, in the next class, we are going to study about the limitations. Okay. Here you can see this is protein layer, and this is our phospholipid, and the force of attraction between them is our van der Waals forces. Yeah. Okay. So in the next class, we are going to study about limitations. Alright. So best of luck. Just go through this model. Then in the next class, we will go to limitation part and then unit membrane model. I hope you get it. Thank you. Bye-bye.